Learning and memory formation are complex processes that involve the dynamic remodeling of neuronal connections, called synapses. To understand the mechanisms that support the plasticity of synapses, we need to be able to visualize the interactions of key proteins involved in neurotransmission at the molecular level. The current imaging approaches available for observing live cells are limited to optical methods, since other ultra-high resolution methods, such as scanning electron microscopy, cannot be used to study living cells. To see molecules in optical microscopy, fluorescent tags known as fluorophores are used as molecular labels. These fluorophores are excited by light to create a fluorescent image. To increase the contrast, confocal microscopy uses a scanning laser to excite the sample and collects fluorescence through a pinhole aperture to filter out the non-focused light. However, diffraction of light waves restricts the light from being focused to a point smaller than 200 nanometers. As a result, optical systems are unable to resolve biological material below this size, making it impossible to see the molecular interactions inside synapses. To overcome this obstacle, researchers from Laval University have developed a novel technique known as Fluorescence Lifetime Imaging Nanoscopy, or FLIN. FLIN combines fluorescence lifetime imaging technique with stimulated emission depletion nanoscopy to measure the protein interactions through a phenomenon known as Forster resonance energy transfer. This combination enables them to visualize and quantify molecular interactions inside the synapse. In order to detect protein interactions, two different types of fluorophores are first introduced to label specific proteins. The interactions between these proteins is then detected through a phenomenon known as Forster resonance energy transfer, or FRET. FRET involves a non-radiative energy transfer from the so-called donor fluorophore to the so-called acceptor fluorophore, when the two molecules become less than 10 nanometers apart. This energy transfer occurs due to the overlap between the emission spectrum of the donor fluorophore and the absorption spectrum of the acceptor fluorophore. To quantify these interactions by FRET, a technique known as Fluorescence Lifetime Imaging Microscopy, or FLIM, is used. When a fluorophore is excited by light, it releases a photon after a certain period of time. This period of time is referred to as the fluorescence lifetime, which may vary between individual fluorophores. In FLIM, the photons released by the population of fluorophores are counted to measure the mean fluorescence lifetime. When FRET occurs, the energy is transferred to the acceptor fluorophore instead of being released as a photon. Since FRET is more likely to occur for donor fluorophores with longer lifetimes, the fluorescence lifetime of the population is shortened during molecular interactions. This allows FLIM to accurately quantify FRET by measuring the decrease in fluorescence lifetime. These lifetimes are color-coded according to their durations, and a false-colored image is then generated to map out the areas of protein interaction. However, even though the combination of FRET and FLIM provides quantitative information about the protein interactions, imaging with standard confocal microscopy is unable to resolve the exact location of the interactions within the synapse. To improve the resolution, the traditional confocal microscopy is replaced with stimulated emission depletion nanoscopy, or STED. In STED nanoscopy, the excitation beam is overlaid with a donut-shaped depletion beam to deactivate the surrounding fluorophores. As a result, images can be generated from emission spots smaller than 200 nanometers, which allows the nanoscale objects to be resolved. By combining the FLIM-FRET technique with STED nanoscopy, Fluorescence Lifetime Imaging Nanoscopy allows for the precise localization and quantification of important protein interactions involved in neurotransmission, enabling a deeper understanding of the mechanisms behind learning and memory formation. This imaging technique provides a novel way to explore the detailed molecular processes that occur within living cells, and it has the potential to impact many disciplines of biomedical sciences.